Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on December 18th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 72nd episode of Matters Microbial. I appreciate every listener and viewer, as well as every email and comment. From time to time, I receive email questions, and I'm happy to answer them. YouTube questions are a little less accessible to me. As we approach the holidays, I hope you can appreciate this image of my micromoss tree in my little laboratory. Yesterday was the final exam for my microbiology course at the University of Puget Sound. I was inspired by something I saw on social media, a hashtag #ThankAMicrobe poster for different microorganisms and why we should be grateful for each. So, I made it an assignment for the final exam. #ThankAMicrobe number 1 was for lactobacillus, which is certainly understandable. A number of students chose this one from just looking around textbooks and the internet. #ThankAMicrobe number 2 was for Streptomyces, the producer of many antibiotics and thus important to fighting disease-causing bacteria. #ThankAMicrobe number 3 was an unusual choice. Oceanoplasma caligoria, isolated from deep-sea corals, and was first described in 2024. I have long believed that using creative approaches in the classroom pays off for both instructor and for student, and these 3 examples illustrate my point. Literally. To be clear, many people use creative approaches with students. I'm just one of many. But imagine how I felt when a quiet student was taken by a comment I made some time ago about how some gliding bacteria secrete a surfactant that reduces surface tension in front of the cells, allowing them to skate forward, sort of like a bacterial slip and slide. Here is a drawing that student handed to me before that final exam yesterday. I will so miss these students. It truly has been a privilege. For many years, and this is relevant to today's topic, I have had my micronauts isolate bacteria from their reusable water bottles to learn about isolation from mixtures, single colonies, colony morphology, streaking, dilutions, plating, and 16S ribosomal RNA identification. Here's a gallery of the kinds of isolates we see in student water bottles. As you can see, they vary in color, colonial morphology, and they even fluoresce from time to time under ultraviolet light. Due maybe to siderophores? A few years ago, a student in my microbiology class named Mariko drew a cartoon referring to these microbes as her buddies, as you can see. Thus, the #WaterBottleBuddies logo was born, designed by Ethan Kokik and seen with this sticker. And for this reason, I am particularly excited to have Dr. Ameet Pinto of the Georgia Institute of Technology on the podcast to discuss the research their team has done investigating the drinking water microbiome and why you should care. Welcome to the Quality Quorum for today, Ameet.
Thank you so much for having me. It's a pleasure and an honor.
Well, I have to tell you in all honesty that you first came to my attention for different kind of science in that you have the world's most amazing cat. And this would be Nessie, a calico. And since I'm trained as a geneticist, I love exon activation and I love seeing these kinds of things. And so I believe you're looking at some beautiful images of Nessie, who has apparently their own Instagram page. Is that true?
That is correct. You can follow Nessie on my.name.is.nessie. And she has quite the attitude.
Well, I think it's a fascinating one. And by the way, people interested in that general topic, when I would teach introductory biology, I would often use a book. It's out of print, but it's worth your time. It's called Cats Are Not Peas. And I'll put a link to it in the show notes. The idea is this author had a male calico cat, and that's due to the XXY, thus that you still have exon activation. And it was a reminder that the rules that we teach in genetics are not absolute by any stretch of the imagination. So I have actually shown many pictures of Nessie to my students over the last few years.
That's fantastic to hear.
So I think as you and I talked a little bit, I've done a certain degree of things on a simple level, looking at the kinds of inhabitants of water bottles that my students have seen. But you have a very different thing that you're doing here, and it has to do with the water, the whole drinking water microbiome. So tell us a little bit about this and why it's important.
Yeah, so one of the first things that we have to do is when we try to look, even try to understand why microorganisms in drinking water are important, we have to take a look at how that drinking water is treated and distributed to us. So if we take a look at the image which is labeled how we treat water, on the y-axis you will be able to see these are arbitrary concentration units over there. And on the x-axis, what you're following is the water that goes from the source. This might be a river, it might be a lake, it might be groundwater. It goes through the treatment system where we use a range of different treatment technologies to treat that water, and then that water is pumped into the distribution system. These are pipes that are buried underground, and we can't see them, but they are delivering that treated water into our homes. And then finally, there are pipes in our buildings, which we refer to as building plumbing, uh, where that water has to pass through. So what happens as this water moves from the source to the treatment system is the amount of substrate that is available for microbial growth, and you can see this in the blue line over there, that starts decreasing. And this is because we're using treatment technologies such as coagulation, flocculation, we're using filtration. And all of this is removing the carbon that's in water, the nitrogen that's in water. It's removing potentially the phosphorus that is in water. Um, and then, right as the water is about to leave the drinking water treatment plant, we add a disinfectant. And this disinfectant could be chlorine, it could be chloramine. Um, and our goal now is to make sure that as the water moves to the distribution system, a certain amount of residual disinfectant is there to prevent microbial growth. And when it gets into our buildings, typically the water sits for long enough that we lose some of that disinfectant residual. So this is the process that we're using to treat our drinking water and deliver it to our homes. What we're doing, essentially, if you take all the different engineering technologies and water management strategies, is we are reducing the amount of substrate that is available for microbial growth. And at the same time, we are increasing the amount of chemical stress in the form of disinfection that these microorganisms are subject to. So, from a biological perspective, we are doing everything we can to remove microorganisms. And it makes absolute sense because we don't want pathogens in our water supply. If you think of surface waters, they are in the open environment. There could be fecal pathogens that might wash off due to rains, for example, or there might be animals that might interact with with these surface waters, and that could also contaminate them. So we want to reduce the microbial load, and that's what we're doing. Now, what makes the drinking water microbiome incredibly interesting is that somehow it survives all of these chemical stressors and starvation and makes it through the treatment plant into our homes. Now, thinking about the drinking water microbiome and how we interact with it, if you take a look at the image called water use, this water that comes into our homes, we are interacting with it in a range of different ways. So this is data that you're seeing from 2016 that was collected by the Water Research Foundation, and this data shows that the average American uses about 80 to 100 gallons of water per day. Not all of that is for drinking. We use water for a range of different purposes, from flushing our toilets to showers to faucets to washing our laundry. So there's a range of different uses. But there are a few different uses where we are directly connected to that water, and that is showering. Those droplets, there is skin exposure, there are bioaerosols that are also formed during that process. We might inhale some of those droplets. Then we drink water, we cook with water. And so these are all different exposure routes from which we are sort of interacting with the drinking water microbiome. And despite what I've said in terms of all these different treatment technologies reducing the microbial concentration, there is actually a substantial number of microorganisms that make it through. So if you take a look at the image titled concentrations, what you'll see is there is close to, so there is, despite what we do in terms of treating our drinking water before it gets to us, there is actually a substantial concentration. It varies from about 1,000 cells to 100,000 cells in every milliliter of drinking water. So if I now take those numbers and I put them in terms of how much water we use and how much water we are exposed to, it adds up quite significantly, right? It's in the millions of cells from drinking water that we are exposed to. So that is why the drinking water microbiome is important, is because we are exposed to it on a daily basis. It is highly resistant, or it can deal with lots of different stressors. And at the same time, there could be potential pathogens within this drinking water microbiome. And that's why we have to kind of control what's in our water supply and manage it.
So it's really interesting that you bring this up on a couple of different levels. I mean, number one, we always make assumptions about turning on the tap and getting potable water, clean water. And in fact, it's an interesting thought, because Snow in England, and I'll put a link to this in the show notes, was one of the first people to demonstrate that you could get the transmission of cholera via the water supply. And there was one particular contaminated water supply in London. I believe they've made kind of a monument there to remind people of that. And it's interesting. A little anecdote that I would like to tell listeners and viewers about is the term oligotrophy. And these are organisms that can live on very, very small amounts of carbon and nitrogen. And I have obviously this is what you're going to find in drinking water. But what I found interesting is when I was first, how would I put this in an elegant fashion, dating the woman who's now my wife. Do you know the large water containers, and there's like a device you put it in and then you can like distribute water? And it was almost empty. So I was going to show how strong I was by moving a new one on there. But I cleaned out the ceramic that was holding that large bottle. And my wife, my lady friend at the time, said to me, "Why are you doing that? It's just water." And I took my finger and I went, and there was all the stuff. This is the challenge for water treatment when you think about it.
Yeah, there are estimates, and now we don't have a really good handle on this because, as you can imagine, the pipes that are buried underground are not easy to access, especially
Right.
not easy to access without contaminating them. When there is a leak, for example, there are estimates that maybe about 90% of the microbial biomass in these drinking water distribution systems are in the form of biofilms. So what we are actually measuring at the tap is a very, very small proportion of the microbial community and microbial biomass. In fact, biofilms have been a high priority area in terms of research for a really long period of time. And coming back to your story, Mark, I clean my cat fountains on a weekly basis. And their cat fountain gets filtered water, and I'm surprised at how much biofilm I have to clean every Saturday morning.
It's not really a surprise to a lot of
Hmm.
us. We think about dental plaque, that's a biofilm,
Mm, yeah,
and it recovers very quickly. But anything that where there's flowing water, it kind of behooves those organisms to glue themselves down.
that's right.
And
Yeah.
and that way the nutrients pass by them.
Hmm.
It's just amazing stuff. So when I was doing these experiments with student water bottles, we would just swab them, very low
Yeah.
tech. And it was interesting because some water bottles would have a high bio load and others a low bio load. And I don't know if it's possible to really tell if that has anything to do with the care and feeding of a water bottle, because that gets personal with people. You know, if you ask them how often do you wash that, they hear that as a
Hmm.
judgment, right?
Yeah.
But this is why it's so cool to have you here to talk about things that we all take for granted every time we turn on that tap.
Hmm. Yeah. And it's remarkable given that that water passes through pipes that are decades old, right? But most of the times, the water that we get is very, very safe. It's fresh, and it's a luxury um, that we often don't appreciate. That we have clean drinking water on demand at the tap whenever we want
It's
it.
it's not shared by a lot of the world. So, you
That's
know,
right.
your own background is in kind of the engineering thing, which we'll come to later. But this is so critical, and it's something I really want listeners and viewers to remember is how much they take that for granted every day. You'll never look at your you'll never look at your tap the same way. By the way, I once had a student who wanted to see if Brita filters, I shouldn't use the trade name, excuse me, whether those filters that one puts in the refrigerator actually did a lot of filtration of microbes. And no,
So,
sorry.
we call them point of use filters. They're really, really good for chemical contaminants, for example. They're helpful for lead if there are issues
Oh,
with
yeah.
lead in the community, removal of that. They are good with disinfection byproducts, for example. But as you said, if there is water, if there is a surface, microorganisms will grow. They might remove the microorganisms that are in the water on one side, but if the other side is has to be wet and it has a surface, microbes will find a way.
I usually say it's first evolved, last extinct. And it's really true, you know, and I have to admit, I have to, in all clarity, I sometimes make fun of what I call swab stories, which is a play on words of sob stories,
Hmm.
where people will swab a surface and say, oh gosh, there are microbes there. Well, yes, of course.
Hmm.
Um, but it's really interesting not to gross people out. That's not my intention, but to let students know the things they take for granted. Like I had students asking me about water fountains, and I said to them, there's a difference between what they're drinking out of and the pipes delivering it. And those are things that you really have to think about.
Hmm. Hmm.
Well, I think that this is a really good start to it. I was fascinated by some of the things. I mean, you have a wonderful website, and I of course will link to that. And any other links that you would like the
Hmm.
listeners and viewers to see, I'm happy to add. But I usually go crazy myself. But it's such a fun topic. I wanted to tell you that when I was looking at, and we'll come to this in a second, to some of the inhabitants in the water, in the drinking water microbiome, I saw lots of organisms that I've seen in the student bottles,
Hmm.
like a lot of Sphingomonas.
Uh-huh.
I've seen a lot of Chrysiobacters. I didn't see that on your list,
Ah,
but a lot of Chrysiobacters. I mean,
interesting.
every year I see those.
Yeah, yeah.
I see Rotobacters. I see all
Sure.
kinds of interesting things. And
Yeah,
then you get the ones like Bacillus that almost certainly have nothing to do with the water,
yeah.
right? Or you'll get things that very clearly were because they were reintroduced as the person drinks.
That makes perfect sense. Yeah.
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 So I think
And
a good place to go ahead. What were you going to
no, no,
say?
please, please, yeah. No.
So how would you go about studying these things? Do you filter the material and then you
Hmm.
isolate? Mhm.
Yeah. So the way we approach our studying the drinking water microbiome is using a couple different strategies. The first one is we are very interested in what fraction of the microorganisms in the sample that we collect have intact cell membranes. So we will use a technique such as flow cytometry coupled with live dead staining to get a sense of, there are 100,000 cells in here, what fraction have intact cell membranes? Because as you can imagine, these microorganisms are under constant chemical stress and likely to have damaged membranes. So that's one area of investigation, is what fraction. And what's surprising is you can actually get lots of great information from that. You can not only get data on total cells, cells with intact cell membranes. You can also, because the dyes that we are using will label nucleic acid, you can kind of start separating high nucleic acid from low nucleic acid cells. So you can start getting multiple layers of information. And there have been very nice developments. These are not from my group. These are from a group in Belgium. Very nice developments in this space where you can analyze that data in a statistical framework to get some metrics that tell you something about the community, such as its alpha diversity or community fingerprint. So that's one tool that we use on a regular basis. It's a very quick technique. It requires expensive instrumentation, which is a flow cytometer, but it's very high throughput. You can run lots and lots of samples very quickly. So that's one way that we do that. And that is to get quantitative estimates of the concentration of microbial cells per milliliter of drinking water. The second technique that we use is now nucleic acid based. So what we'll do, as you can imagine, the concentrations are quite low. So we will typically filter this water through different types of filters depending on our need. So one common example is a 0.22 micron pore size filter, and we will filter water through that. Depending on the concentration, we might filter 1 liter, we might filter 10 liters. But anything above that, it's very difficult to get through a 0.2 micron filter. And that's because we are not just talking about microorganisms. There is other stuff in this water, humic acids, for example, that are in the water as well. There can be some particulate products of corrosion from these pipes that might be in the water sample. And these filters tend to clog up. Now, if you need to filter higher volumes of water, we'll go to what's called an ultrafilter, where we can filter. I was actually just last Friday filtering about 350 liters of water in my bathroom for an experiment that my student wanted to do in terms of DNA extraction. So we will use these large volume concentrators. And so it depends, the concentration of microorganisms matters and what we want to do with that sample. So as an example, if the concentration of cells is 1,000 cells per milliliter, and if it's now 100 cells per milliliter, that's a tenfold difference. So we might need to go from 1 liter of water to 10 liters of water, or from 10 liters to 1,000 liters. So that's once that the microbial cells are concentrated onto the filter membrane, we will then extract their DNA. And we have to be quite careful over here, because if you're dealing with a low biomass environment, contamination or likelihood of contamination is very, very high. So our students in my research group go through quite a bit of training on how to handle these samples. They go through extensive QA/QC procedures in terms of reproducibility of their DNA extractions. We will even do some very quick and dirty mass balances. We measured 1,000 cells in every milliliter. Let's assume that genome is X size, 3 million base pairs. How much DNA did we get? And let's back calculate. So we try to do these exercises just to make sure that there is a good QA/QC in place. And at the same time, we will also run some of these blank filters through the extraction process, and then take them the entire way through whatever analysis we want to do. Now, once that DNA is extracted, and I'm talking about quite low levels of DNA, if we get 50 nanograms per microliter of DNA, we'll be super happy. But oftentimes we're talking about 0.2 or 0.3 nanograms per microliter in the DNA extract. And we have certain rules about how that has to be above the background level and all of that. And I can share more details on that. Once that DNA is extracted, now it depends on what we want to do with it. What is the research question at hand? If we are looking for specific type of microorganisms, Legionella pneumophila, for example, or Pseudomonas aeruginosa, then we will do digital PCR. We will go target a specific gene of a well-known organism and use some of these quantitative assays to get a sense of how many gene copies in every microliter of DNA extract. Now, I'm saying microliter of DNA extract because there is an extraction bias over here. We cannot translate gene copies directly to how many cells in a milliliter of water. Through lots and lots of experience, we have a sense of what our typical recoveries are. So we can, in terms of the extraction bias, so we can do some ballpark estimation, but it's very difficult to extrapolate that. So if it's an organism that we know, we want to quantify, then we will go and we will do digital PCR or quantitative PCR. The second method is now if we are interested in knowing its taxonomy. What is the identity of a diverse group of organisms in that particular sample? In that particular case, we will go after the small subunit ribosomal RNA gene, either the 16S rRNA gene or the 18S rRNA gene. Now remember, drinking water is not just bacteria. There are amoeba in drinking water. There are fungi that can be present in drinking water. So if you want to go from bacteria and archaea to eukaryotes, you're going to have to use a different marker gene. And once we amplify that particular marker gene, we will then either do Illumina sequencing, which is a short read approach, and this we will go after a very, very specific hypervariable region of the small subunit rRNA gene. Or more recently, we've been playing around with both MinION sequencing and PacBio sequencing, and both of these platforms allow for the full length of these small subunit rRNA genes. The full length is about 1500 to 1600 base pairs for bacteria and archaea, and 1800 and higher sometimes for eukaryotes. And what that does is it gives us information about the identity of the microorganism. If it's a short region that we've sequenced, then it might tell something about this family that this organism or that sequence belongs to, sometimes at the genus level, rarely to the species level. And that is why when we go after the full length of the 16S or the 18S gene, we start getting species level resolution of what's in our water supply. Now, the thing to remember over here, this doesn't tell you what the microorganism can do. It only tells you their name. And, considering the huge amount of diversity that's out there, if we see a Legionella pneumophila gene sequence, then we can be fairly sure what that microorganism can do or its risk. But if we see, for example, Spingomonas, especially if there is not a genome in the genome database that's closely related to it, then we don't know what it can do. So we then take it a step forward, and what we'll do is we'll do metagenomic sequencing. And this means now we take all the extracted DNA and we take a shotgun approach to it. Now we are not targeting any particular gene, but we will sequence whatever is in that pool. And there is quite sophisticated bioinformatic approaches now to actually take that data and assemble it in ways where we can start putting genomes together. You can think about it as if trying to piece together a puzzle of small pieces without actually having a guide of how those pieces fit together. Thanks to a lot of great bioinformaticians out there, these tools are highly, highly accessible. So that's what we'll do. And once we have a genome, now we can annotate those genes and see what functions are within those group of microorganisms. And that is highly, highly informative. In fact, we have recently released a resource, and this is a file dwgc.pdf, where we have collated these genomes from a range of different studies that have been done globally. And we've created, I believe, the first drinking water genome catalog. That is a publicly available resource. It's clearly not all the microorganisms in our water supply system globally, but it does capture some of the key microorganisms that are seen across the globe. And these are highly curated genomes for the community to use in their own research, update, and share as you would like. So these are the range of different techniques that we use to look at the drinking water microbiome. Culturing is an important part, has traditionally been done. water utilities will routinely do heterotrophic plate counts using R2A agar plates. This is quite routine at water utilities, and um, it gives you a sense of what this culturable cell concentration is. Those numbers rarely correlate very well with flow cytometry, for example, because of course there is a certain bias when it comes to culture. I'm sure there is a bias when it comes to flow cytometry as well. So that is also a very, very common technique that's used.
Oh, this is actually so wonderful on so many levels because these are things we've talked about a little bit in prior episodes. We introduced some ideas about metagenomics last time. You did a wonderful explanation there. I mean, to give listeners and viewers an idea, like, what if I were looking at microbes? I'm not doing this, I'm just suggesting it. If we looked at microbes in plastic versus metal water bottles, you
Ah,
might ask the question, do you have organisms on the plastic ones that have genes associated with catabolizing some of the, for lack of a better term, leachate that all types of plastic water bottles give?
mm-hmm,
Or metal resistance genes, perhaps?
mm,
That kind of thing. And you have that ability now. So it's not so much as who goes there, who's in your water, but what are they
yeah,
doing?
yeah.
And that's the
What, what
important part. Go ahead.
what are they capable of doing? Because a gene is present, it
Thank
may
you.
or may not be expressed. One of the things that we've struggled quite a bit, and that's because we are in such a low biomass environment, is actually getting RNA that we can work with to look at what genes are being actively transcribed. This becomes super challenging. There are some studies out there where they have sequenced RNA from drinking water, but these studies are typically from systems that do not use a disinfectant. So there are countries around the world where they do not maintain a chlorine residual in the distribution system. This is true in the Netherlands, this is true in Switzerland, this is true in Denmark, maybe parts of Germany, where they distribute water without any disinfectant, and the cell concentrations there tend to be higher. The microbial communities tend to be much more diverse. And so there are studies from some of these countries, even in Finland, where they can get enough RNA to do sequencing. We've tried, we've not been successful, but we keep trying.
I have to laugh because last year at this time I was in Iceland. It
Oh,
was my
cool.
25th anniversary with my wife. And I don't know what it is about Iceland, and I loved it. It's a wonderful place. Everybody in Iceland has a set thing that they say about their drinking water. It's like they
Okay.
all learned the same words. And it is, this water is the purest and best water on earth, filtered through many tons of volcanic basalt. And it's like, it's
Uh-huh.
all they're like, is it like the Pledge of Allegiance in school or something? And it makes me wonder, what is that drinking water like? Who's in there? Right?
Yeah.
And I mean, it's a
Yeah. Good.
fascinating idea. Yeah.
Yeah. No, it's a great question. I don't know who's in there because I don't think I have seen a sequencing based study from Iceland of drinking water. So if someone's out there and wants to send me a water sample from Iceland, I'd be happy to sequence it.
And it's interesting too because all in Reykjavik anyway, everything is geothermally heated, and that water comes out just under boiling. And my wife said to me, this is a peak Doc Martin moment. I wanted to somehow swab up in those pipes and bring a sample back, but I didn't because I wasn't supposed to.
Oh, okay.
Oh, you know
Um,
how it is. You don't go to another country and take samples and stuff like that. I could have, but I
yeah,
didn't.
yeah, yeah.
But it does go to something that I want listeners and viewers to really appreciate is that we tend to be reductionist in the way that we look at microbes in general. They're on plates, they form single colonies, they're in a culture tube in liquid. This isn't how they live. Nor even when they're in biofilms, they're not by themselves. So in essence, what you're talking about is an ecosystem within the pipes of our water
That's
system.
correct. Absolutely.
And the
So,
things that
and
we go ahead.
yeah, and this ecosystem is actually shaped by engineering. So what's remarkable to me is that you can take a water sample from Atlanta and you can take a water sample from Hong Kong. And as long as they are disinfecting and they're using similar treatment technologies, you'll likely find very, very similar microorganisms. Doesn't matter that they are so far away. So I have, I believe, a plot which just says occupancy, Racaniello occupancy. So what we did over here is we took a look at all those genomes that we had put together, and we estimated for each genome how many samples it was seen in and what its abundance was in that sample. And on this plot, what you can see is on the y-axis is the mean relative abundance of each of these genomes. Each genome is a blue dot. And on the x-axis is how often it was detected. And these are all systems that disinfect with chlorine or chloramine. And what you can see is there, once you go above a 0.5% relative abundance and 33% detection frequency, there are about 5 or 6 genuses that are seen in many, many water samples. So it's disinfection, which is one of these key, it's an incredible treatment technology. It's an incredible engineering intervention, life-saving. There is lots and lots of literature out there how drinking water disinfection essentially, played a huge role in, and continues to play a huge role in protecting our health. It selects for certain groups of organisms. And that's what engineering intervention does. We want to get high quality, safe water to all consumers. And so we implement tested and optimized and validated treatment technologies to make sure that that water is safe. And in doing so, we are also shaping the ecosystem that's in our pipes.
I mean, it's not a surprise. Anything that we mess with is going to be altered by what we do. And like what I often say is the hand of Darwin is on all that lives. And so anytime that we change an environment, this takes place. And this brings to mind something, and in no way am I going, I fear I sound flippant too often, and I don't mean to. I'm very serious about this. Now, Norm Pace, who did a lot of some of the early environmental samplings, and you're probably familiar with the old paper where he found out what was living on his shower curtain, right? But my favorite, and this is relevant to you, is the Showerhead study. Are you familiar with that?
Yes, yeah. Is it from Norm Pace's? Or I know that Noah Fearer's group also has had a study recently as well. Yeah.
And so it turns out that there were some of these, and by the way, I don't recommend it, but I love it because it's me. When you get an old shower head and look inside, you can see the biofilms, and those are awesome.
Yeah.
I would love a 3-dimensional image of what's in there, but you have to have properly marked cells to do that, but you get the idea.
So there is, I'll refer your viewers to a study that was led out of EWAG by Dr. Frederick Hamas's group. And his group has been looking at not shower heads, but shower hoses. These are plastic hoses inside that long shower line, and it has thick, rich biofilms. And he's got some beautiful images, SEM or ESEM images, and he put some false color on them. You go to his website; they're gorgeous. And I think they might even be in some papers of his.
You need to send me that link if you would be so kind. But what was interesting about this, and this is relevant to what you just said, is that there were some areas that had more mycobacteria than other areas in the shower heads. Now, I want to be very clear, just when you get the supposed genus, it doesn't mean it's really a particular member. In other words, if I get bacillus, it doesn't mean Bacillus anthracis, that kind of thing, right? So you got a lot more mycobacteria in some regions than not, and those were the areas that used a lot more chlorine than others because the mycolic acid in the outer membrane protected those organisms against the extra chlorine. So, just as you said, modifying the environment shaped the community.
That's correct. Absolutely. Absolutely. It's not almost, it's exactly that. It's an environmental filter, and that's the engineering intervention. Yeah.
No, I think that's wonderful. we've had a wonderful talk so far about this. Are there some specific projects that you'd like to talk about that you're doing in your group?
Sure. So right now, we have a few different projects going on in the drinking water space. One of them is funded by the US EPA under their National Priorities Program. So this is an interesting thing that water utilities deal with on a regular basis, and that is they have to disinfect to prevent microbial contamination. But when you add that chlorine to drinking water or chloramine to drinking water, it reacts with the organic and inorganic stuff that's naturally occurring in our water supply to produce what is called disinfection byproducts. And there is a host of different disinfection byproducts. And what we do know that some of these disinfection byproducts can have adverse health impacts, whether it's genotoxicity, whether it's I'm not an expert in the disinfection byproduct side of things. So water, and these are regulated, by the way. Many of these are regulated. Water utilities have to measure these disinfection byproducts. They have to be below a certain acceptable, below a certain level of risk, for example. But water utilities are constantly navigating this. How do we maintain a certain disinfectant residual to keep the microbial concentrations down, while at the same time make sure the disinfection byproducts stay below a certain threshold? However, the way those decisions are made, there is a certain science behind it, but the trade-off between reducing concentrations of microorganisms and formation of disinfection byproducts, that is not very quantitative and it can vary quite a bit from system to system. So as part of this particular project, what we're trying to do is we are working with water utilities across the country and with our collaborators both at Georgia Tech, at University of North Carolina, Chapel Hill, at Northeastern University, and our research institute that's quite near us, the Water Tower Institute. We are working with water utilities to try to understand what the exact trade-off is. How often do you see disinfection byproducts in your systems? And how are they correlated to chlorine concentrations? How are they correlated to microbial concentrations? And in particular, Legionella pneumophila. Legionella pneumophila has emerged as one of the top waterborne disease concerns, especially in the United States. I believe there was a paper that came out earlier this year that said 85% of all waterborne infections in the United States are from Legionella pneumophila. Now, this is not, these are not a lot of infections, but they're still quite significant. This is a bug that probably doesn't affect people with healthy immune systems,
Yes.
but it will immunocompromise the elderly. It can be quite a deadly pathogen. We also don't have a very good sense of how widely distributed Legionella pneumophila is in our water system. So this is an important project that we're working on. This project started last year and is going to continue on for a few more years. Hopefully by the end of this project, we'll have a sense of how often do you see Legionella pneumophila in our water systems, and how is its presence or absence linked to disinfection and disinfection byproducts. So that is an important project that's going on. There is a large team involved. The second drinking water project that's quite interesting that we have going on is on a new microorganism that we just discovered. This microorganism, and this is a plot that says Raskinella_1. And what you're seeing over here on the right is in blue, the circles, is a particular genus that's called UBA4765 on the x-axis. And there is a big blue data point right up top. Turns out that this data point represents a genome that is found in close to 80 to 82% of water systems globally. Surprisingly, it was not characterized. It was UBA4765. It's not in culture. There is a genome out there in the genome databases, but this turns out to be very, very widely distributed. So we are heavily focused on this microorganism because we have assembled this genome independently from many different systems. We have annotated this genome. We know it's a biofilm former. We know it has telltale signs of disinfection resistance. uh, we know it can degrade some of these disinfection byproducts that are formed in drinking water systems. We know that it has quorum sensing genes, and so it's a fascinating microorganism. Very, very likely, if you're getting disinfected drinking water, that you have encountered this microorganism or encounter it on a daily basis, just because how widely it's distributed. And so focus quite heavily on this microorganisms as well over the next few years. We want to get this microorganism in culture, and I think I'm collaborating with one of your previous guests, Dr. Paul Carini, on trying to get this microorganism in culture. Of course, Paul is an expert at getting things that are difficult to get in culture into culture. So we're collaborating with Paul on that particular aspect. But we really think that this particular microorganism will be the model microorganism for the drinking water microbiome or drinking water ecosystem. I was very inspired by some of the papers that came out of Joe Handelsman's group about a model community. So
Yes.
her group put together Thor, these a simplified community for the soil ecosystem. I would love to do something like that for the drinking water ecosystem, and so that we can understand intricate things about biofilm formation that we might not, because these biofilms are, because we don't have microorganisms that reflect the drinking water ecosystem in culture. So UBA 4765, we have now renamed it Droskinella chloroaquaticus. We want to get that microorganism in culture, create this model community for the drinking water microbiome. And it's not just about knowing, using that model community to try to understand how the ecosystem functions in the drinking water distribution system. It's also about technology testing. We can now use this model community to test filtration technologies, to test disinfection technologies, to test UV treatment technologies. So we can actually do these technology testing with microorganisms that actually reflect the drinking water ecosystem, and not E. coli, for example. Even though E. coli is fabulous, it's a wonderful bacterium, but it's not representative of the drinking water ecosystem.
No, and I mean, one of the things that I talk about with my students are the deadly centrisms. And one of them is coli-centricity. And I can't tell you the number of times even really smart scientists will think that a new organism does things exactly the way the organism they're familiar with
Hmm.
does that. And
That's
it's
right.
not true. I think it was Bill Metcalf, a colleague of mine from a long time ago, who said about phenotypic assays, you know,
Mm,
that's not how the microbe looks at it at all,
that's right.
right? And this idea of a community is really important on all kinds of levels. I mean, I would even argue that this is relevant to astrobiology, um, because I think this idea of a low population community. I use this term, and please forgive me because I'm sure I'm using it incorrectly. High bioload and low bioload is what I tell
Uh-huh.
the students. And the fact is, these oligotrophs are used to low population densities in most cases. I'm
Mm,
particularly interested in more and more. We tend to be so reductionist. We want to look at one organism. It's how I was trained, right? It's not how it works at all. And so when people were studying biofilms, I used to say, well, biofilms really aren't monocultures of anything. It's okay.
mhm,
I'm looking at two different organisms.
mhm,
Well, that's not the answer either. You do have to start. And that's why Joe Handelsman's work was so important with Thor. And even with this four-part system, you have such complexity,
mhm.
right? Which is just amazing. And
It's incredible. It's
yeah,
incredible. Yeah.
yeah. And so I would not be at all surprised to learn about they're all hanging on the inside of these pipes, doing their best to make a living
Yeah.
there in the dark, right?
Yeah. That's right. Yeah.
So if you've learned, Amit, that some microbes in the water bottle ecology, not water bottle, so you've learned that some of the drinking water microbiome constituents, some of those organisms appear to become resistant to some of the sanitizing agents that are being used or disinfecting agents that are being used. Are those genes being undergoing horizontal gene transfer to other members of the community? Because that would
Mm,
seem like something that you might expect.
that's a great question. So first, I should add a caveat over here. And the caveat is that these are genes that we have annotated in a genome for a microorganism that does not exist in culture.
Yes.
So just because we attribute a function to it does not mean first that the function does what it does once we get it in cultivation. So that is one of the reasons this top microorganism, Roscanella chloroquarticus, is so important, because we can see signs of potential disinfection resistance. But whether it is actually how resistant it is, we don't know yet until we get it in culture. The second mechanism that actually contributes to resistance has very little to do with disinfection resistance, but it has to do with physics. Once you form biofilms, if the microorganisms can shelter within those biofilms, disinfectant cannot penetrate deep into those biofilms. So it's essentially just biofilm sheltering. And these biofilm formation genes are so widely distributed that, they might. So I don't have a good answer for why stuff is not getting horizontally transferred. Um, and maybe that happens and we don't know about it, or but it's not just about disinfection resistance. There are other ways in which this ecosystem gets shaped, and the other way is reducing the amount of carbon that's available for growth. So we are removing so much. So these microorganisms might have other ways of making a living that are so central to their metabolism that maybe those getting horizontally transferred is not that likely. So I'm not saying it's not happening or it doesn't happen, but I also can't be sure that those particular traits that we identified from a 3.5 million base pair genome, those are the only ones that are responsible for why a microorganism is abundant in the drinking water ecosystem.
And I'm so impressed by this work. And every time that people say that they've kind of gotten to name an organism, I'm all excited, because that must be a great feeling.
Yeah, it was fantastic, especially because this is going to be I quite an important organism for the drinking water community moving
Oh
forward.
yeah.
Yeah.
No, I think this is fabulous on so many levels. And so this has been just a fabulous session for that reason. And what I'd like to do in the time remaining is for you to talk a little bit about your path, because your path, one thing I've said over and over again to listeners and viewers, is there's not one path to microbiology. There's lots. And would you care to share your experience?
Sure. You know, I was trained as a chemical engineer. I like mass balance. I like energy balance. I like chemical reactions. So when I finished my undergraduate degree in chemical engineering in Mumbai, I was not so passionate about the profession. I was not so passionate about working in pharmaceutical, even though it's fantastic, or petrochemical, even though that's great. But I took a course in my elective in my fourth year last year, which was called Environmental Science and Pollution Control. And that textbook is still there somewhere back there. And then I realized that I could take the same principles of chemical engineering and use them to solve environmental challenges, environmental problems. And so I started pursuing environmental engineering. I did my master's in environmental engineering, I did my PhD in environmental engineering. And my master's and PhD, for the most part, were very process oriented. You know, um, uh, Biology is at the heart of wastewater treatment. That's what I did my PhD in, wastewater treatment. These are large, huge biological reactors that treat billions of gallons of waste on a daily basis. But they're all modeled around the metabolism of individual microbial groups, right? And to me, that was quite amazing that somehow what we learned from microorganisms can scale up and deliver function at these absolutely large volumes. And that at some point during my PhD, it hit me that, yeah, I'm a chemical engineer and these are chemical reactions that are packed inside membranes. And so I like to think about microorganisms as bioreactors. And that's how sort of, little tiny bioreactors. And that's how sort of That was my step into microbiology during my PhD. I also happened to read during my PhD some fantastic work that came out of Tom Curtis's group and Bill Sloan's group. Tom Curtis is a engineer, ecologist, fun person, crazy person at Newcastle University, and Bill Sloan is a hydrologist turned ecological modeler, stochastic modeler at the University of Glasgow. And I started reading their papers, and they were coming at these engineering bioreactors from the point of view of of ecologists. For me, that was absolutely fascinating. So my love for biology is rooted in looking at, or microbiology is rooted in looking at these little tiny bioreactors that are surrounded by other tiny bioreactors that form a microbial community. That's how I sort of stumbled into microbiology from sort of a process engineering background.
You know, it's interesting because when you talk to most people about what it is to be a microbiologist, they'll have kind of a negative view about the name and the term germ. Don't get me started. I have a whole speech I give on that one. But the important part is that there are lots of different paths. Mine was from symbiotic communities.
Mm.
That's what I was most interested in. And other people, and there's nothing wrong with being medically oriented with this stuff at all. There's nothing wrong with it. But that wasn't what my interest was. And I wish I had a more eloquent term for it, but some people use the term a big tent for a lot of people with different interests, like working together. So let's make it that way for microbiology, but we'll call it a big petri dish. And that's kind of where we're at. That's where we need to have this place where we can all kind of work together. And I honestly believe, and Curtis and Sloan are great examples, that sometimes the most interesting ideas come from people outside the discipline. You know, because you could be a formal microbial ecologist and you might miss all that. You see what I mean? And so bringing these
Mm-hmm.
new ideas in are so critical. And that's one thing I love about doing the podcast is I get to hear from lots of different people, lots of different points of view.
Yeah.
So do you have any final pieces of advice for people who might be listening or viewing? I mean, I would like to emphasize something that you said is that we have pure, good water out of our taps.
Yeah. I think drinking water, clean drinking water, is a luxury um, that we should not take for granted. We should make sure that it stays clean. We should make sure that we invest in our water infrastructure. Just because we don't see it doesn't mean it does not exist. And for us to get clean drinking water on a daily basis, we need to make sure that our governments, from cities to federal, are investing in our water infrastructure.
There's a statement that people make, and I don't mean again to be flippant, but they'll say some areas, please forgive this, are sexy. That is to say, people like hearing about them. But honestly, the nuts and bolts of everyday life are far more relevant to everybody.
Hmm.
And I can't think of something more central in this country right now than the knowledge that you turn on the tap and you have water that you can drink.
Exactly. And it's not just about drinking. I remember in December 2022, we had a big freeze in Atlanta and we had some water main breaks. And I was cooking on Christmas Day, and in the afternoon we lost water. And we did not get water for 3 to 4 days. The amount of inconvenience, because these were pretty large water main breaks and that had, took time to fix. The inconvenience of not having water is incredible. You can't flush your toilets. You
Mhm.
can't wash your hands. You can't. So it just water touches that clean drinking water touches our lives in so many different ways and makes our lives convenient in so many different ways. It's absolutely incredible that we have that in our homes.
So Amit, there's kind of a meme that goes around about drinking water in different locations around the world, safe versus unsafe. And I don't know how much of that is a Western point of view and how much isn't. How much of that do you think has to do with the starting place cultural practices or what's accepted?
Yeah, great question. Um, so water is typically treated in very, very similar ways across the world, where there is a centralized treatment facility. Many places around the world do not have the infrastructure to have centralized treatment facilities. So that's one thing that I would put out there. So if there is not a central treatment, municipal treatment facility, then you know the standards of treatment are going to be are going to be quite variable. The second thing that I would say is in most places in what I would say the developed countries, not just the Western world, in Japan, in China, in Australia, in United States, we have continuous water supply. That means our pipes are always under pressure. It's always flowing. And what that pressure does is if there are leaks, keeps contaminants out because the water flows outside. In many parts of the world, either it's an electricity issue, maybe it's a governance issue. It might not be a technology issue, but it could be an electricity issue. So I grew up in Mumbai, and we used to get water 2 hours in the morning and 2 hours in the evening, and we would pump that water up to a storage tank that was sitting on the roof, and the water would sit stagnant there for most of the day, and we would use it. So we had to boil the water, we had to filter the water before we used it. So that is an important aspect, is the nature of intermittent water supplies. And a huge portion of the world is on intermittent water supplies. Might not have to do with the water infrastructure at all. It might have to do with the electrical grid, for example.
Yes.
Um, Intermittent water supply increases the likelihood of can increase the likelihood of contamination. So that's one thing. So now when I travel to India, I drink bottled water. Even though I grew up over there, I know that my likelihood of getting exposed to something that my GI tract cannot handle is higher. Even though my parents use an RO system to filter that water, I will still try to stick to bottled water. So I think this is it's not an engineering thing. The engineering to the technologies that are out there are quite well understood. It might be a combination of governance, infrastructure, how expensive it is to sometimes treat water, and whether there is whether there are the resources to actually have that kind of treatment system in place. So those are the reasons. And the other thing that I will add to that is once a pipe goes in the ground, it's very difficult to pull it up and replace it. It's a very expensive project. So even in parts of the world that might now initially might have been on intermittent water supply, but now have continuous water supply, that microbial community that was developed 10 years ago, 15 years ago, 20 years ago is still hanging around. So I think that's one of the key reasons where um, if you travel to parts of the world where the water supply is intermittent, either have it makes sense to be cautious.
Thank you very much for that. I thought the intermittent water supply is something that had not occurred to me. So thank you very much. Well, I think that's a good place to kind of start to segue at the end. I want to thank you for your time, Ameet. Not only are you the caretaker of Nessie, which is just about the coolest thing ever, you've given a really wonderful introduction to listeners and viewers about the centrality of clean water, the technologies behind it, the interface between microbial ecology and engineering that I think everyone will find fascinating. And I want to thank you for your time.
Thank you so much for having me on this podcast. I've watched so many episodes, and it's great that I get to be part of one.
Well, you certainly have been a wonderful guest. Thank you so much.
Awesome. Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Ameet Pinto is the Carlton S. Wilder Associate Professor in Civil and Environmental Engineering at the Georgia Institute of Technology in Atlanta, Georgia. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.